exceeded $8 billion, which is more than the total investments of any country that shares India's border, like China. But the point is different. The point is India is worried about these policies, about what China may do with investments, and that is why the policy has happened. But China says that these are discriminatory practices, and we should to treat investments from different countries in different ways is not equitable at all. But this was done. Nobody will say this, of course. The Indian government will make no such comment on it. But the general understanding is that this was done keeping in mind China's practices, which are not entirely, India believes, our board.
Absolutely. So and it's not that the investments have been blocked. All that has been done in terms of modification in the policy is that these countries that share their borders with India will have to take an approval from the government prior to making any such investment, or prior to in fact changing the title or of ownership or transfer of ownership. Okay, Srinjo, before you answer that, let me in fact go across to strategic affairs expert Sushant Sarin joining us on the broadcast. Like I said, sir, or of ownership or transfer of ownership.
Oh, public. Hello friends, just mark your attendance so that we know that you're now available. Keep marking your attendance, send in a hi or a thumbs up so that we know that you are here. Good. So I can see all of you are signing in. Yesterday we had lots of problem with the buffering. We went back to the technical team and we have probably discovered what was the issue with the buffering, since the internet connection was quite good. So we are hoping that today we don't face that problem. We have tried to resolve it on the technical end, and since the internet speed is anyway good, we are expecting that today the buffering issue should not occur. Fingers crossed. Let's hope that there is no buffering issue today. Is the voice okay now? Is the voice okay now? Just let me know if the voice is okay now. Initially the mic was away from me, so the voice might have been a bit low, but I presume it should be okay now. In addition, it would be great if you are able to use a pair of earphones or a pair of headphones, because a lot of time the phone speaker or the laptop speaker may not be enough if you are using fans or AC, or if you're sitting on the outside room. So it is suggested that you use a pair of headphones preferably. If you don't have a pair of headphones, then a pair of earphones will do, but definitely use some hearing aid to get the best audio output from your device, whether it is a laptop or either it is a phone or a tab. Please try to use a hearing aid, whether it is a headphone preferable. If you don't have a headphone, then the earphones should also do. Okay, so what I'll do, we'll do a quick recap of what we had discussed yesterday, and then we'll move on from there. Yesterday we had started the discussion on the glycolysis. Unfortunately, we could not complete it because of the problem with the buffering. So let's quickly take a quick look at what we had discussed yesterday and carry on from there. So we were discussing the glycolysis yesterday. We were discussing the glycolysis yesterday. I told you the other name for glycolysis is the EMP pathway. The other name for glycolysis is the EMP pathway, right? The Emden-Merhoff-Parnas pathway. It has two phases. The preparatory phase, which is also known as the investment phase, because ATP is consumed in this phase, and then we have the payoff phase where ATP is actually generated. In addition, the glycolysis is of two types depending on availability of oxygen. We call it the aerobic and the anaerobic glycolysis. So two types of glycolysis, the aerobic and the anaerobic glycolysis. So talking about the location where the glycolysis occurs, we mentioned certain tissues where exclusively anaerobic glycolysis occurs, and these were tissues where either the mitochondria is not available or oxygen is not available because they are avascular tissues. So RBC doesn't have mitochondria, and the cornea, lens, and subepithelial layer of skin and GIT, they are avascular tissue. Exception was the outermost layer of cornea, which can get some atmosphere from the atmosphere through the tear film. Then there are certain tissues which show both aerobic and anaerobic glycolysis. We call it the mixed glycolysis. These tissues are the CNS and the renal medulla. The CNS and the renal medulla. All other tissues are exclusively aerobic under resting conditions. Please note the qualifier here. They are exclusively aerobic under resting conditions. Under special conditions, the metabolism may change. All right, but exclusively aerobic under resting conditions. For example, we had seen yesterday, if you're doing exercise, the muscles may have the anaerobic glycolysis going on. Okay. Then we talked about the compartment. All of you know that glycolysis occurs in the cytoplasm. The substrates are commonly the hexoses, most commonly glucose, but the glycogen is also a potential substrate. The end products for aerobic and anaerobic glycolysis are obviously different. For aerobic glycolysis, from one molecule of glucose, what you get two molecules of pyruvate and two molecules of NADH. In case of anaerobic glycolysis, one molecule of glucose is able to give you only the two molecule of lactate. Now, the rate limiting enzyme, you must remember in some guides, wrong answer is given. The answer is phosphofructokinase 1, also known as the PFK1. It is the most important rate limiting enzyme of glycolysis. What are the targets for regulation? Another important question. Targets for regulation glycolysis, they are PFK2, which is under hormonal regulation, PFK1, which is under the allosteric regulation. When you talk about allosteric regulation, we must mention the activators and the inhibitors. The activators for PFK1 are AMP, the fructose 6-phosphate, and fructose 2,6-bisphosphate, which is itself generated from PFK2. The inhibitors are ATP and citrate. We also mentioned that the fructose 6-phosphate is an example of feed forward activation. It is an example of feed forward activation. Then we said hexokinase shows the feedback inhibition. by the glucose 6 phosphate. Once the glucose 6 phosphate is present in larger amount, it will start inhibiting the hexokinase. The glucokinase concentration, or you can say the total amount, is increased by the presence of insulin. Lastly, pyruvate kinase itself is also activated by insulin. So here you will note most of the enzymes that are having a regulatory function, the PFK1, the glucokinase, the pyruvate kinase, are somehow affected by insulin, but hexokinase is not affected by insulin. Okay, then we talked about energetics. Energetics was a bit tricky, and I told you we have different energetics for aerobic and anaerobic glycolysis. And in addition, for different tissues under anaerobic and aerobic conditions, we get different output. For RBC, we said because of the Rappaport Lubering shunt, we'll see this, we are able to get 0 or 1 ATP. We are able to get 0 or 1 ATP, whereas for most tissues it is normally 2 ATP. For most tissues it is normally 2 ATP. In muscles, if anaerobic glycolysis occurs during the exercising, and at that time the glycogen will be the fuel, in that case we end up with a 3 ATP. In that case we end up with a 3 ATP. Similarly, when you talk about aerobic glycolysis for different tissues, for example, brain, where the shuttle which is operating is the glycerol 3 phosphate shuttle, we only get 5 ATP. But for kidney, for liver, where malate aspartate shuttle is operating, we can get up to 7 ATP. Alright. At this point, we started talking about the reactions. We said glucose converted to glucose 6 phosphate by the enzyme hexokinase. Another enzyme can be the glucokinase. Hexokinase or glucokinase will convert the glucose to glucose 6 phosphate. They are transferases, they'll transfer the phosphate from ATP to the glucose. For this, magnesium is also required. The glucose 6 phosphate is quickly isomerized to fructose 6 phosphate. This is a reversible reaction by the enzyme isomerase. The full name is phosphohexose isomerase. Fructose 6 phosphate is then converted to fructose 1,6 bisphosphate by the enzyme PFK1. Again, because it is a kinase, ATP is consumed and magnesium is required. Out of these three, first and third are irreversible reactions. Second is the reversible reaction. Together, they constitute the investment phase. Together, they constitute the investment phase. Okay, we have a question. Fructose 1,6 bisphosphate, does it inhibit PFK1? No, fructose 1,6 bisphosphate does not inhibit the PFK1. Fructose 1,6 bisphosphate does not inhibit the PFK1. Allosteric regulation, will do this in detail when you're doing the enzymes, but simply speaking, allosteric regulation means certain molecules can get attached to enzyme, thereby modifying its activity. The activity may be increased, the activity may be decreased. The activity may be increased or it may be decreased. Okay, so we have completed the investment phase. At this point, the fructose 1,6 bisphosphate is acted upon by aldolase. Aldolase is of two types, aldolase A and B, which splits the fructose 1,6 bisphosphate into the dihydroxycetone phosphate and glyceraldehyde 3 phosphate, both of which are 3 carbon compound, or they are trioses. They are trioses and they are freely interconvertible by the enzyme triose phosphate isomerase. Freely interconvertible by the enzyme triose phosphate isomerase. The glyceraldehyde 3-phosphate is subsequently quickly converted into 1,3-BPG by the action of glyceraldehyde 3-phosphate dehydrogenase. Remember, this is also a reversible reaction. Inorganic phosphate is consumed for this reaction. In the process, NAD is converted to NADH. NAD is converted to NADH, resulting in formation of 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate acted upon by the 3-phosphoglycerate kinase. Remember, this is a reversible reaction where ADP is converted to ATP. Magnesium is consumed. This is what we call the substrate level phosphorylation. This is what we call substrate level phosphorylation, where ATP is synthesized during a chemical reaction. ATP being synthesized during a chemical reaction, we call it the substrate level phosphorylation. The 3-phosphoglycerate then converted to 2-phosphoglycerate by the phosphoglycerate mutase. The 2-phosphoglycerate then converted to phosphoryl pyruvate by the enolase. Phosphoryl pyruvate then irreversibly, please note, irreversibly converted into pyruvate by the enzyme pyruvate kinase. This is the second place where ATP is generated. So it is the second example of substrate level of phosphorylation in glycolysis. So in two steps we find the substrate level phosphorylation in glycolysis. One is by the 3-phosphoglycerate kinase and second is by the pyruvate kinase. Under anaerobic conditions, the pyruvate can be further converted to lactate, which is a reversible reaction catalyzed by the enzyme lactate dehydrogenase, in which NADH is converted back to NAD plus. NADH converted back to NAD plus. All right. Now here I had started talking about the glycogen as the source for glycolysis, and we had mentioned that the glycogen can generate glucose 1-phosphate, where the donor for phosphate is pyridoxal phosphate, written as a PLP. This is a pyridoxal phosphate, and glucose 1-phosphate can get converted to glucose 6-phosphate by the mutase enzyme. Phosphoglucomutase will carry out this conversion. The first enzyme was the glycogen phosphorylase. We'll do this reaction in detail later, don't worry. So glycogen to glucose 1-phosphate and glucose 1-phosphate to glucose 6-phosphate. Please note, in this process ATP has not been consumed. If you carry out the conversion of glucose 6-phosphate from glucose, in this direction ATP is consumed. But if you carry out the conversion from glycogen to glucose 1-phosphate, ATP is not consumed. That is why when glycogen is the fuel for anaerobic glycolysis, we have one more ATP. One extra ATP becomes available, and we had written it was 3 ATP. If you recall, we had mentioned it was 3 ATP. Second thing to note, all the reactions from this point onwards will occur twice. I had mentioned this previously also. All the reactions from this point onwards will occur twice, simply because we have two trioses. So all these reactions will occur twice. Conversion of glyceride 3-phosphate dehydrogenase action by making the 1,3-BPG, then subsequently right up till the pyruvate. Everything will occur twice. Now in the RBC there is a slightly separate shuttle which is available, which may be used. It is not necessary that it is always used. It may be used, and that is resulting in conversion of 1,3-BPG first into 2,3-BPG, and this 2,3-BPG then getting converted to 3-phosphoglycerate. It is then getting converted to 3-phosphoglycerate. This is what I had told you is known as the Rappaport-Leu-Behring shunt. The Rappaport-Leu-Behring shunt exclusively seen in the RBCs. The Rappaport-Leu-Behring shunt. And when the Rappaport-Leu-Behring shunt operates, please note again, we don't get ATP. This ATP which was being formed here will not be formed when you use the Rappaport-Leu-Behring shunt. That is why in RBC the net gain of ATP can be 0 or 1 depending how many trioses enter the Rappaport-Leu-Behring shunt. Depending on how many trioses are entering the Rappaport-Leu-Behring shunt, the net gain may be 0 or 1 ATP in case of RBC. The PLP stands for pyridoxal phosphate. It stands for pyridoxal phosphate. It is the active form of vitamin B6. It is the active form of vitamin B6. We'll do this when we are doing the vitamins. When we are doing the vitamins, I'll come to this pyridoxal phosphate. It is a very important intermediate in large number of metabolic pathways, particularly as the donor of phosphate. Particularly as donor of phosphate, it is a very important coenzyme in large number of reactions. The last point that I'll make here, see what is happening. 2 ATP consumed in the preparatory phase, right? 2 ATP twice generated in the payoff phase. So net gain is 2 ATP plus 2 NADH for aerobic glycolysis. This is what will happen in most of the tissues. We had seen the difference in RBC because the Rappaport Lupering shunt, instead of 2 we are getting the 0 or 1. In case of the muscles, instead of 2 we are getting the 3 ATP. Now this 2 NADH will go to mitochondria only in aerobic conditions. It will go to mitochondria only in aerobic conditions. In anaerobic conditions, it will be used up to convert the pyruvate back to lactate. Okay, we know this. It will be used up to convert the pyruvate back to lactate. So the NADH goes to mitochondria. We say that it goes to mitochondria, but the fact is it cannot enter the mitochondria. So what happens? The NADH gives its reducing equivalent to one of the two shuttles which are present at the inner mitochondrial membrane. At the inner mitochondrial membrane, two shuttles are present where the reducing equivalent is handed off by the NADH. The two shuttles which are available are the glycerol 3 phosphate shuttle and the malate aspartate shuttle. These are the two shuttles which are available to transport the reducing equivalent of NADH in the matrix of mitochondria. Now, when glycerol 3 phosphate shuttle is used, there is some loss of reducing equivalent, and instead of NADH, FADH2 is generated. So from 2 NADH you get 2 FADH2. But when you're using the malate aspartate shuttle, there is no loss of energy, and a result again 2 NADH will get generated. 2 NADH will get generated. Both of these will generate ATP with the help of the electron transport chain. Both of these will generate energy with the help of electron transport chain. But what is happening here? The FADH2 will be able to generate 1.5 ATP, whereas the NADH is able to generate 2.5 ATP. So from NADH 3 ATP when glycerol 3 phosphate shuttle is there, and from NADH 5 ATP when the malate aspartate shuttle is there. Previously in the tissue we had seen the gain of 2 ATP. So depending on which shuttle you are using, you end up with either a 5 ATP or 7 ATP. Depending on which shuttle is being used in the tissue, you end up with either 5 ATP or the 7 ATP. This 2 ATP net is coming from the reactions in the cytoplasm we had seen previously. So I hope this clarifies the points related to the uh, amount of ATP being generated in the aerobic as well as anaerobic glycolysis. The aerobic and anaerobic glycolysis. Very briefly, I'll talk about the glycogen as fuel. See what is happening when glycogen is fuel. It will generate the glucose 1 phosphate. We'll see this again when we are doing the glycogen metabolism. We'll see this again. The enzyme catalyzing the reaction is the glycogen phosphorylase. The glycogen phosphorylase will catalyze this reaction, and the pyridoxal phosphate will provide the phosphate. The phosphate is coming from pyridoxal phosphate and not from ATP. It is not coming from ATP. ATP is not involved. ATP is not involved here. Subsequently, this glucose 1 phosphate is readily converted into the glucose 6 phosphate. It is readily converted into the glucose 6 phosphate, which will then participate in rest of the glycolysis. Now, if you generate this glucose 6 phosphate in glycolysis from glucose, in this reaction ATP is consumed. But if you use glycogen as fuel, the ATP is not consumed. That is why we had said when glycogen is the fuel, net gain is plus 3 ATP. The ATP that you have saved here, it will increase the net gain. But in case of glucose, the net gain was 2 ATP. This would be very simple. Reason being, what is happening here? Consumption only 1 ATP. Production 4 ATP. Right? So net gain is 3 ATP, whereas here the consumption is still of 2 ATP and production is 4 ATP. I hope this clears up the glycogen as fuel. This clears up the glycogen as fuel that we are talking about. Okay, where are we? Yeah. Okay, so glycogen as fuel. I would say the enzymes involved in rapid liver shunt are not important, so please don't go into that because there is no end to the details that we can have. But I would rather not give those enzymes to you. It will just clutter up the space in your mind. Please don't go into the details of the Rappaport Libering shunt. Only thing you need to know is Rappaport Libering shunt is operating in the RBC. It generates the very important intermediate, which is the 2,3 BPG. It generates the very important intermediate, which is the 2,3 BPG. This is the only thing that you need to remember. Everything else is superfluous information, not required. Not required. Everything else is superfluous. Please don't go into details. There is no end of details. All right, so let's not be greedy. Let's focus at the end result. End result is we have to do those topics important for the exam. No point in going into the details. Remember, Rappaport Libering shunt exclusively seen in the RBCs. The intermediate 2,3 BPG is important. As a result of our Rappaport Libering shunt in RBC, net gain of ATP is less. It can either be 0 or it can be 1 ATP. Can anyone tell me the importance of this intermediate 2,3 BPG? What is the importance of this intermediate 2,3 BPG? The importance of this intermediate 2,3 BPG. I'll wait for your answers. Quickly send me what you think is the importance of 2,3-PG. Why in the RBC the 2,3-BPG is being generated? What is the need of the Rappaport bearing shunt inside the RBC? It is not operating anywhere else. Why in the RBC we need the Rappaport bearing shunt to generate the 2,3-BPG? What possible role is 2,3-BPG playing in the RBC? Again, this is a crossover from physiology. I have said it previously. What is the role of 2,3-PG inside the RBC? Why do we need it? Send in your answers. I'll be back in a minute. Okay. I can see lots of answers coming in. Oxygenation of hemoglobin, affinity towards hemoglobin, oxygen dissociation prevents from oxidative stress, oxygen affinity, loss of oxygen binding affinity. Very good. So quickly, I'll mention the role of 2,3-BPG. Very briefly, I'll mention it. 2,3-BPG shifts oxygen hemoglobin dissociation curve to right. In very simple words, makes dissociation favorable at low partial pressure of oxygen. In simple words, if you want to say, it makes the dissociation favorable at low partial pressure. That is why if you are in a place where there is a low partial pressure of oxygen, for example, you are at high altitude, you have gone to Ladakh to visit that lake in the Three Idiots, the Pyongyang lake. There the partial pressure of oxygen is very low. So to make the dissociation favorable, the RBC will start making more and more of 2,3-BPG. More and more of trioses will be diverted towards the 2,3-BPG synthesis. Result net gain will be zero. But at sea level, the requirement of 2,3-BPG is comparatively less. So some of the triose goes through the Rappaport bearing shunt. Some of the triose goes through the normal glycolysis. The net gain of ATP in this case is 1 ATP. Alright, so how much 2,3-PG is formed depends on what is the partial pressure of oxygen. At low partial pressure, we require more 2,3-PG. At normal partial pressure, one less amount of 2,3-PG will be enough to favorably affect the oxygen hemoglobin dissociation curve. Okay, so that was the main points about the glycolysis. Only extra thing that you need to know are the three enzymes which catalyze the irreversible reaction. The three enzymes which catalyze the irreversible reaction, I'll highlight them. Pyruvate kinase, pyruvate kinase, the PFK1, the pyruvate kinase PFK1, and hexokinase oblique glucokinase. These are the three enzymes. They are the three enzymes which catalyze irreversible reaction. These are the three enzymes which catalyze the irreversible reaction, number 1, number 2, and number 3. The three reactions, the three enzymes which catalyze the irreversible reaction: hexokinase oblique glucokinase, the PFK1, and a pyruvate kinase. Another very important conceptual point that I will highlight here is about irreversible reaction. When I say irreversible reaction, what does it mean? Does the backward reaction occur? Is it possible to carry out the backward reaction? Yes or no? For irreversible reactions, can backward reaction occur? Yes, no. Please send in your answers. What do you mean by irreversible reaction? Does it mean the backward reaction does not occur? Yes, no. Backward reaction occurs? Yes or no? What is your answer? I'm waiting for your answers. What do you think happens in an irreversible reaction? Backward reaction cannot happen. Yes, no. For irreversible reactions, backward reaction can happen, but they must be catalyzed by a different enzyme. For irreversible reaction, same enzyme cannot catalyze backward reaction. So when I say backward reaction, it does not mean that backward reaction cannot happen. Backward reaction can happen, but it will only happen when you have a different enzyme. The same enzyme will not be able to catalyze the backward reaction. Backward reaction will happen, may happen. I will not say will happen, may happen, but you will need a different enzyme to catalyze the backward reaction. The same enzyme will not be able to catalyze the backward reaction. So please keep this point very clear in your mind. When we say irreversible reaction, this means for the backward reaction you'll require a different enzyme. I hope this point is clear, and this also brings us to the summary of glycolysis. What we had seen till now, what has happened, and that is the conclusion of glycolysis. At this point, at the end of glycolysis, if we are talking about the aerobic glycolysis, if we are talking about aerobic glycolysis, we are left with two molecules of pyruvate, right? So now we need to know what happens to this pyruvate that you're getting at the end of glycolysis. What is the fate of the pyruvate that we obtained at the end of glycolysis? This pyruvate can get metabolized in the cytoplasm. One possible fate, it gets metabolized in the cytoplasm. Second possible fate, second possible fate is it enters the mitochondria. Please note, this entry will be facilitated by a transporter. This entry will be facilitated by a transporter, and now it will enter the matrix of mitochondria. In the cytoplasm, again, it can get converted to two things. Some of them we have already seen. The pyruvate can get converted into lactate. This we had seen, the conversion being catalyzed by the lactate and dehydrogenase. The cytoplasm conversion to lactate catalyzed by the lactate dehydrogenase, a reversible reaction. Another possible fate is the pyruvate can be converted into alanine by the enzyme alanine transaminase. By the enzyme alanine transaminase, it can be converted into alanine. Please note that both the reactions are freely reversible. Both the reactions are freely reversible. Thank you. Okay, so both the reactions are freely reversible. Moving on to the mitochondria, let's see what happens inside the mitochondria. In the mitochondria, again, the pyruvate has two possible fates. More commonly, the pyruvate is irreversibly converted into acetyl coenzyme A. It is irreversibly converted into acetyl coenzyme A by the enzyme pyruvate dehydrogenase. This is the more common fate of the pyruvate in the mitochondria. But sometimes, depending on the requirement, the pyruvate can also get converted again irreversibly to oxaloacetate. It can get converted to oxaloacetate by the enzyme pyruvate carboxylase. Please note carboxylases. Carboxylases, when we say carboxylases, they will require the vitamin B7. It requires vitamin B7. We'll see this later, and that is biotin is required for carboxylation. In addition, for this reaction, ATP is consumed. For this reaction, ATP is consumed. Alright, the two molecules oxaloacetate and acetyl coenzyme A have different fates. They have very different fates. The acetyl coenzyme A is likely to enter the Tca cycle. This is the common fate of the acetyl coenzyme A. It will enter the Tca cycle. Beyond the Tca cycle, it can have some different fates, but it will enter the Tca cycle. The oxaloacetate, on the other hand, more commonly, it will be directed towards what we know as gluconeogenesis. It will be directed towards what we know as the gluconeogenesis. Alright, so pyruvate in the cytoplasm, it can be converted to lactate and alanine. Different enzymes are there, the lactate dehydrogenase and alanine transaminase. Both these reactions are freely reversible. In the matrix of mitochondria, pyruvate can again get converted to products by irreversible reactions. More commonly, it is converted into the acetyl coenzyme A, but sometimes, depending on requirement, it can also get converted into the oxaloacetate. It can also get converted into the oxaloacetate. Okay, the oxaloacetate is commonly directed towards the gluconeogenesis, whereas acetyl coenzyme A will enter the Tca cycle. The acetyl coenzyme A will enter the Tca cycle. Okay, so at this point we come to the conclusion of glycolysis. Let's take up another metabolism which is primarily occurring in the cytoplasm, few reactions in the mitochondria, and that is the gluconeogenesis. Okay, that is the gluconeogenesis. So let us quickly talk about the gluconeogenesis. Before I go to gluconeogenesis, any questions so far? Any questions that you want to ask from glycolysis? It is an open house now. Next, we are going to be talking about gluconeogenesis. So, open house for questions. Any doubts, any queries, any part you want me to repeat from glycolysis? Today, we are going to continue till 11. It will be a 3-hour session. So, I'll request you to request your family members to give you some grub so that you can sit for 3 hours. Just like I have got some grub, make sure you have something to keep you sustained for 3 hours. In the meantime, if you have any queries, any doubts, any part you want me to repeat again, please send them across. No questions? Okay. Question: aerobic lactate formed? Answer is no. Lactate will be formed only in anaerobic conditions. Lactate will be formed only in anaerobic conditions. Any other question? Any other question from glycolysis? No other questions? Okay. So let's move on to the gluconeogenesis. If there are no more questions, we are moving on to gluconeogenesis. First, what do you mean by gluconeogenesis? It means synthesis of glucose from non-carbohydrate sources. Gluconeogenesis is a synthesis of glucose from non-carbohydrate sources. Now, this is an energy-intensive process. Therefore, it occurs only in fasting, starvation. In other words, when glucose is low. Alright, synthesis of glucose from non-carbohydrate sources. When glucose starts going down, when the glucose store in the body starts going down, that is when we carry out the gluconeogenesis. So here what I'll do, I'll quickly do the checklist, and then I'll show you the 3 stages or the 3 reactions of glycolysis which need to be bypassed. The 3 reactions of glycolysis which need to be bypassed to generate new glucose from different molecules. Okay, so what we're going to do next is the checklist. The checklist. So remember, in the checklist, the first thing that we mention is the location or the organ where the pathway occurs. Which are the organs where new glucose can be formed? I expect you to know the answer to this question, so I'll wait for you to give me the answer. Tell me the location, the organs, the tissues where new glucose can be formed. I'll give you a hint. There are only two of them. The hint is there are only two tissues or two organs where gluconeogenesis can occur. What are the two organs or tissues where gluconeogenesis can occur? The gluconeogenesis occurs only in two tissues, as I mentioned. The two tissues are liver and kidney. The two tissues are liver and kidney. Gluconeogenesis does not occur anywhere else. Does not occur anywhere else. Liver is good. Liver is good. Anything else? Mm, very good. Very good. So in a checklist, first point, the location organs where the gluconeogenesis occurs. The answer was liver and kidney. Second, compartment. Two compartments are involved in gluconeogenesis. Two compartments are involved in gluconeogenesis. Anyone would like to take a guess? Which are the two compartments? I've already mentioned the two compartments. They are first mitochondria, but mainly it is the cytoplasm. Most of the reactions of gluconeogenesis also occur in the cytoplasm. All right. So compartment, mitochondria and cytoplasm. Mitochondria and cytoplasm are involved in gluconeogenesis. Now comes the very frequently asked question: what are the substrates for gluconeogenesis? What are the substrates for gluconeogenesis? Five well-known substrates for gluconeogenesis. For humans, only four of them are actually applicable, but you should know the five. You should know the five substrates for gluconeogenesis. I'll wait for you to give me some names and then I'll start writing the substrates for gluconeogenesis. Remember, the most important substrate for gluconeogenesis is always the amino acids. And among the amino acids, the most important substrate is the alanine. What else? Besides amino acids, what else? Anything else besides the amino acids? Yes, that is lactate. In addition to lactate, pyruvate. In addition to lactate, pyruvate. Oxalate is not considered a substrate, rather it is an intermediate. Oxalate is not considered a substrate, rather it is an intermediate. What else? Then we have glycerol, and then we have propionate. This propionate is coming from odd chain fatty acids. So if odd chain fatty acid is given in the answer, that is also correct. Obviously, only the propionate part of the odd chain fatty acid, the last three carbon residues are able to participate in the gluconeogenesis. The end product we already know is glucose. Okay, so where it is occurring, which compartments are involved, what are the substrates, and what is the end product? This we have done till now. Okay, I'll again highlight. Remember, the most important substrate for gluconeogenesis is alanine. There is one special condition where some other substrate becomes more important, and that is after severe exercise, or in other words, after anaerobic exercise, the lactate becomes the more important source. The lactate becomes the more important source, but only after severe exercise. Otherwise, alanine is the most important substrate for gluconeogenesis. Okay, now here we have to know 2 points: the rate limiting enzyme and the target for regulation. The rate limiting enzyme and a target for regulation, they are the same enzyme. The rate limiting enzyme and target for regulation are the same enzyme, and the answer to this is fructose 1,6 bis or di. Both names are acceptable. Fructose 1,6 bis or diphosphatase. Fructose 1,6 bis or the diphosphatase, the rate limiting enzyme as well as the target for regulation. Rather than talking about target for regulation, more commonly what is asked is how or what regulates. Okay, how or what regulates. So one of the common questions is most potent stimulator of gluconeogenesis. What is the most potent stimulator of gluconeogenesis? Besides that, what are the other activators of gluconeogenesis, and which is the inhibitor of gluconeogenesis? Remember, we are talking in terms of hormones. We are talking in terms of hormones. You have to tell me which is the most potent stimulator for gluconeogenesis, which are the other activators for gluconeogenesis, and which hormone is the inhibitor for gluconeogenesis. We are talking about hormones. Glucagon is definitely the activator of gluconeogenesis, but it is not the most potent stimulator. Glucagon and epinephrine are definitely the regulators, and just you have said the insulin is the inhibitor. But which is the most potent stimulator? I have not yet got the correct answer. the most potent stimulator for gluconeogenesis? The answer is glucocorticoids. For example, cortisol. The glucocorticoids are the most potent stimulator. They are not the others, they are the most potent stimulators. They are the most potent stimulators. The okay, so that is about the regulation. That is about the regulation. If we talk about energetics, energetics is obviously variable, and it depends on substrate. It depends on substrate for the reaction. Two molecules of pyruvate finally giving rise to one molecule of glucose. The energetics is 4 ATP, 2 GTP, and 2 NADH. In other words, 35% more energy consumed than what is synthesized in glycolysis. All right. Okay. Any questions so far? Because now I'm quickly going to show you the reactions, or you can say the pathway for the gluconeogenesis. If you have any doubts, any questions so far, please tell me. Else, I'll quickly show you the reactions for the gluconeogenesis. In the meantime, I'll quickly summarize what I have said till now. The gluconeogenesis is the synthesis of glucose from non-carbohydrate sources. It is a synthesis of glucose from non-carbohydrate sources, meaning this will occur only in fasting or starvation when the glucose is low. This will occur only in fasting or starvation when the glucose is low. Then we made the checklist. The gluconeogenesis occurs only in two tissues, the liver and in the kidney. The compartments involved are the mitochondria and the cytoplasm. The five substrates which participate in gluconeogenesis are amino acids, that is most important of them being the alanine, most important being the alanine. The lactate is most important after severe exercise or anaerobic exercise. The other substrates are the pyruvate, the glycerol, and the propionate. Propionate as a substrate comes from odd chain fatty acid. The last three carbon of the odd chain fatty acid that is left out of metabolism can contribute to gluconeogenesis. The end product is obviously glucose. The rate limiting enzyme and the target for regulation are the same, and that is the fructose 1,6 bis or the diphosphatase. We'll see where this enzyme is involved in the process very soon. More importantly, we should know how or which molecules participate in the regulation, particularly in terms of hormones. We said the most potent stimulator for gluconeogenesis are the glucocorticoids, particularly the cortisol. The other activators include glucagon and epinephrine. Other activators include glucagon and epinephrine. Inhibitor is the insulin. Inhibitor is the insulin. Talking about energetics, obviously it will depend on the substrates because we have different substrates. We have different substrates, therefore obviously different energetics will be there. If we start from pyruvate, if we start from pyruvate, two molecules of pyruvate giving rise to one molecule of glucose, we end up consuming 4 ATP, 2 GTP and 2 NADH. We end up consuming this, so that the considering that 2 NADH can give rise to 5 ATP, considering that 2 NADH can give rise to 5 ATP, in all 11 ATP are consumed. 4 ATP plus 2 GTP plus 5 ATP. GTP is equivalent to ATP. The GTP and ATP are equivalent, so that makes 4 plus 2 plus 5 ATP, together 11 ATP. 11 ATP are consumed in converting two molecules of pyruvate to one molecule of glucose. We will see these steps and where the energy is consumed when we are doing the backward reaction. When we are doing the backward reaction, then we'll quickly see where this energy is consumed and what are the steps where the energy is getting consumed. All right. So let's quickly go to the process whereby we are going to convert the two molecules of pyruvate into one molecule of glucose. Once you see the process, the energetics should become more clear. Okay, starting with pyruvate. Remember, this part is occurring inside the mitochondria. This part is occurring in the matrix of mitochondria. This part is occurring in the matrix of mitochondria. We already seen the pyruvate will get converted. It will get converted into oxaloacetate. It will get converted into oxaloacetate. This is an irreversible reaction. The enzyme catalyzing the reaction is pyruvate carboxylase. Pyruvate carboxylase, and in the process ATP is consumed. ATP is consumed and vitamin B7 is required for the carboxylation. Vitamin B7 is required for the carboxylation. Okay, so pyruvate converted into oxaloacetate. The oxaloacetate is reversibly converted into malate by the enzyme malate dehydrogenase. By the enzyme malate dehydrogenase. I'm not going to write the energetics here, although it should be something like this. The NADH getting converted into NAD plus. The malate will move out. through a shuttle, the malate will move out now into the cytoplasm. Now it will move out into the cytoplasm. In the cytoplasm, this malate is converted back to oxaloacetate. It is converted back to oxaloacetate by the same enzyme, the malate dehydrogenase, malate dehydrogenase, and the NAD is converted back to NADH. So no net gain or loss. At this stage, there is no net gain or loss. Here we don't have any net gain or loss. The oxaloacetate is then irreversibly converted into the phosphoenol pyruvate, commonly known as a PEP. Commonly known as PEP. Okay. Here the enzyme catalyzing the reaction. The enzyme catalyzing the reaction is PEP carboxykinase. It is a PEP carboxykinase and it consumes GTP. Remember, it is a kinase, it is transferring the phosphate and it uses the GTP converted to GTP. One very important point to note here, the pyruvate can never get converted to phosphoenol pyruvate directly. In glycolysis, we had seen the phosphoenol pyruvate getting converted to pyruvate in the cytoplasm, but the backward reaction, pyruvate to phosphoenol pyruvate, cannot be catalyzed. We don't have any enzyme for catalyzing the backward reaction from pyruvate to phosphoenol pyruvate. We must go through this pathway. Pyruvate going to the mitochondria where it is converted first to oxaloacetate and then malate. Malate comes out, gets converted to oxaloacetate, and then the phosphoenol pyruvate is formed. Remember, from pyruvate you cannot generate the phosphoenol pyruvate directly. There is no enzyme to catalyze the conversion of pyruvate to phosphoenol pyruvate directly. Okay, this prevents the futile cycles. Mean you are doing the glycolysis and immediately it is being used back for gluconeogenesis. Does not happen. Okay, till now what we have seen till now, two unique enzymes, the pyruvate carboxylase and the PEP carboxykinase. These two are together used to bypass the enzyme. They are used to bypass the enzyme pyruvate kinase. Remember, pyruvate kinase was one of the irreversible enzymes. The enzyme catalyzing one of the irreversible reactions was pyruvate kinase. We have bypassed the pyruvate kinase using the pyruvate carboxylase and the PEP carboxykinase. In this process, for every pyruvate, 2 ATP equivalent have been used. One here and one here. Remember, ATP and GTP are equivalent. So for every pyruvate, we make use of 2 ATP till now. What happens now? From phosphoenol pyruvate, we get the 2-phosphoglycerate. 2-phosphoglycerate will give rise to the 3-phosphoglycerate. 3-phosphoglycerate will give rise to the 1,3-BPG. 1,3-BPG will give rise to the glyceraldehyde 3-phosphate, and the glyceraldehyde 3-phosphate will then get converted to fructose 1,6-bisphosphate. Till now everything was reversible. That is why I am not going into the details. I'll quickly mention the reactions. The PEP to 2-phosphoglycerate reversible, 2-phosphoglycerate to 3-phosphoglycerate reversible. If you go to glycolysis, you will see this. The 3-phosphoglycerate to 1,3-BPG is reversible. 1,3-BPG to glyceraldehyde 3-phosphate is reversible. Glyceraldehyde 3-phosphate to DHAP is readily interconvertible, and the DHAP and glyceraldehyde 3-phosphate can combine to form the fructose 1,6-bisphosphate. All of this was reversible. What you have to note is, when you are doing the reversible part, when we go from 1,3-BPG to glyceraldehyde 3-phosphate, 3-phosphoglycerate to 1,3-BPG, at this stage in the backward direction, ATP will be consumed. In the glycolysis, ATP was generated, so now you are going in the backward direction, so ATP will be consumed. Similarly, when you are going from 1,3-BPG to the glyceraldehyde 3-phosphate, the NADH which was generated will now be consumed. Okay, you can see this, and all of this will occur twice. All of this will occur twice, means here you can see 1 ATP, 1 ATP. Here you can see 1 GTP. Here you can see one more ATP, and here you can see 1 NADH. Right? All of this will be consumed twice, because we are also making use of the DHAP, so everything will be used twice. All right, so that's why I said 4 ATP, 2 GTP, and 2 NADH. That is from where we get the values 4 ATP, 2 GTP, and 2 NADH. Now remember, the conversion of fructose 1,6-bisphosphate from fructose 6-phosphate, this was an irreversible reaction catalyzed by the PFK1. It was catalyzed by PFK1, and in which ATP was consumed. Right? This is what we had seen in glycolysis, and magnesium was required. But the backward reaction is catalyzed by obviously a different enzyme. Remember, we had said the irreversible reaction cannot be catalyzed in the backward direction by the same enzyme. We must have a different enzyme, and the enzyme in this case is fructose 1,6-bis, or the diphosphatase. Now please note, the enzyme here is phosphatase. A phosphatase will result in the release of inorganic phosphate. This phosphate will not be trapped. So when you're making the fructose 1,6-bisphosphate, ATP was consumed, but when we are making the fructose 6-phosphate, ATP is not generated. In this backward reaction, the ATP is not being formed. The phosphate is being released in the free form, inorganic form, by the phosphatase. Phosphatase will release it in the free form. The fructose 6-phosphate to glucose 6-phosphate was a freely reversible reaction. Glucose 6-phosphate, if you remember, by the enzyme phosphohexose isomerase, and the conversion of glucose to glucose 6-phosphate from glucose was not a freely reversible reaction. It was not a freely reversible reaction. What happened? It was not a freely reversible reaction. What happened? Something wrong with our system? Okay. It is not a freely reversible reaction catalyzed by the hexokinase or the glucokinase. Hexokinase or the glucokinase. And we had seen for this reaction also ATP was consumed, magnesium was required. The backward reaction is again catalyzed by the enzyme glucose 6 phosphatase, and again because we have the phosphatase, the phosphate is released as inorganic phosphate. The phosphate is released as inorganic phosphate. So at these two steps where you might have expected that some ATP will be regenerated, no, the ATP is not regenerated. The phosphate is liberated in the form of inorganic phosphate. It is not trapped. The irreversible reaction PFK1 in this case is being bypassed by the fructose 1,6 bis or the diphosphatase, and the enzyme hexokinase oblique glucokinase is being bypassed by the glucose 6 phosphatase. It is being bypassed by the glucose 6 phosphatase. So I'll quickly go through the reactions and then we'll summarize some important points for the gluconeogenesis. We'll quickly summarize some important points for the gluconeogenesis and then you can ask your questions. Something has happened here. Okay. Okay. Let's summarize. We are starting the whole process from a pyruvate. We are starting the whole process from a pyruvate, right? Pyruvate first converted into oxaloacetate by the enzyme pyruvate carboxylase. Please note this reaction is occurring inside the mitochondria, in the matrix of mitochondria. The oxaloacetate is reversibly converted to malate. Malate goes into the cytoplasm where it is converted back to oxaloacetate. So whatever energy gain or loss is occurring in between, we are ignoring it. NADH consumed here and regenerated here. So no net gain. We are not going into that. There is no net gain. Okay. The oxaloacetate in the cytoplasm. Is then converted into the phosphoenol pyruvate. It is then converted into the phosphoenol pyruvate by the enzyme pep carboxykinase. Again, this is an irreversible reaction. Again, this is the irreversible reaction. So together they will bypass the enzyme, the pyruvate kinase. Together they will bypass the enzyme pyruvate kinase. Alright, together they will bypass the enzyme pyruvate kinase. The phosphoenol pyruvate reversibly gets converted to 2-phosphoglycerate. We had seen in glycolysis. I'm not going into details. Then to the 3-phosphoglycerate, then to 1,3-BPG, and then to the glyceride 3-phosphate, which combines with DHAP to give us the fructose 1,6-bisphosphate. These are all the reversible reactions of the glycolysis. So I'm not writing the enzymes and the details. What we need to note is when the 3-phosphoglycerate will get converted to 1,3-BPG, the ATP which was initially generated in glycolysis will be consumed. Similarly, the NADH which was generated in glycolysis will be consumed. The DHAP and glycerol 3 phosphate will combine to form a fructose 1,6 bisphosphate, meaning now we are making use of two trioses. Therefore, all the energetics has to be calculated as twice. All the energetics has to be calculated as twice. The two combine to form the fructose 1,6 bisphosphate. The fructose 1,6 bisphosphate is then converted to the fructose 6 phosphate by the enzyme fructose 1,6 bis or the diphosphatase. The fructose 1,6 bis or the diphosphatase. In the process, inorganic phosphate is released. The fructose 6 phosphate is reversibly converted to the glucose 6 phosphate, which is acted upon by the glucose 6 phosphatase. Inorganic phosphate is released and the free glucose is formed. The free glucose is formed. So this is the summary of the gluconeogenesis. I'll just quickly mention the irreversible reactions or enzymes of glycolysis, how they are bypassed, how they are getting bypassed. Okay, PFK1. PFK1 is bypassed first by pyruvate carboxylase and then by PEP carboxykinase. I'm sorry, this is not PFK1. This was pyruvate kinase. I'm so sorry. This was pyruvate kinase. The PFK1 in turn is being bypassed by the fructose 1,6 bis or the diphosphatase. Fructose 1,6 bis or the diphosphatase. And lastly, hexo oblique glucokinase is bypassed by glucose 6 phosphatase. So these are the four enzymes which will bypass the irreversible reactions of glycolysis. They will bypass the irreversible reactions of glycolysis. When we start the process with lactate, mostly coming from the muscle, and this lactate will go to the liver, mostly sometimes to kidney, where it undergoes gluconeogenesis. That will be the Cori cycle, not from pyruvate. Obviously lactate will then get converted to pyruvate, and the pyruvate will finally give rise to the glucose. Okay, so the conversion of lactate to glucose is what we call as the Cori cycle. When alanine gives rise to glucose, we don't call it the Cori cycle, we call it the alanine glucose cycle. We call it the alanine glucose cycle. So I have miniaturized the screen so that you can take a look at the reactions. Only 4 reactions you have to keep in mind, for which I have highlighted the enzyme in the box. The remaining reactions are the reversible reactions of glycolysis, so you don't need to remember them again. There is no need to memorize them again. They are just the reversible reaction of glycolysis. Anyone wants to take this question? Tanuja has asked us, why is it occurring only in the liver and kidney? Why is the gluconeogenesis occurring only in the liver and kidney? Why not everywhere else? Why only those two tissues where the gluconeogenesis is occurring? Anyone who wants to take this question? I'll give you 15 seconds and then I'll write the answer. The answer is the process limiting enzyme. Glucose 6 phosphatase. It is present only in liver and kidney. It is present only in liver and kidney. When we say process limiting, this means whether the process will occur or not. Means if this enzyme is not there, the gluconeogenesis cannot be completed. The gluconeogenesis cannot be completed because we will get stuck at the glucose 6 phosphate step. We will not get the free glucose if the enzyme glucose 6 phosphatase is not there. All right, but the main enzyme which is regulating the rate of reaction, which is the main rate limiting enzyme, is the glucose 6 phosphatase. All these enzymes which we have made in the box, which are irreversible, they have some rate limiting action, but we have to answer the most important rate limiting enzyme. So the most important rate limiting enzyme can only be one, and in this case for gluconeogenesis it will be the fructose 1,6 bisphosphatase. Okay, I'll briefly go over the energetics once again. I can see some questions about the energetics are still persisting, so I'll quickly talk about the energetics once again. Storage location. From pyruvate to oxaloacetate, 1 ATP. From oxaloacetate to phosphanol pyruvate, 1 GTP. From 3-phosphoglycerate to 1,3-BPG, 1 more ATP. And from 1,3-BPG to glyceride 3-phosphate, 1 NADH. 1 NADH. This is for 1 glyceride 3-phosphate. This one also comes from glyceride 3-phosphate, so this is the second one. So that's why we need to multiply it by 2. And remember, GTP is equivalent to the ATP. It is equivalent to ATP. 1 NADH is equivalent to 2.5 ATP. So in other words, what you get is 5.5 ATP into 2, which is 11 ATP. This is what we had mentioned. That 11 ATP are consumed. See, everything is getting consumed. This is minus, 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 and therefore net 11 ATP are consumed if we start from 2 molecule of pyruvate. All right. Any questions from gluconeogenesis? Anything you want to ask me from gluconeogenesis? We have completed the topic of gluconeogenesis. We made the checklist, which you must remember. If you are able to remember something from this reactions that I have shown you, very good. But minimum that you should remember is the checklist that we have made. I also talked to you about the energetics. I talked to you about the irreversible reaction or enzyme glycolysis and which enzymes in gluconeogenesis are responsible for bypassing them. What exactly do you mean by the Krebs cycle, and what is the alanine glucose cycle? These are some of the important points and that you should keep in mind. One person asked why only in liver and kidney. The answer is the glucose 6 phosphatase is present only in the liver and kidney. That is why the process cannot occur anywhere else. Any questions you want to ask me? Any questions that you want to ask me? Okay. See, I'll rewrite these values here. If you start from pyruvate, if you start from pyruvate, in that case, what is happening? Minus 4 ATP, minus 2 GTP, and minus 2 NADH. But if you convert 2 lactate into 2 pyruvate, if you convert 2 lactate into 2 pyruvate, the NAD plus will get converted back to NADH. This is a gain. We have gained 2 NADH. So you have to add 2 NADH. So in this case, the answer becomes minus 4 ATP minus 2 GTP. If you consider it equal to ATP, in that case, the answer becomes minus 6 ATP. Okay. Okay, so we have. I hope this energetics is clear when we are talking about the Cori cycle. In that case, when you're starting from lactate, to make glucose, 6 ATP are consumed. From 2 lactate to make glucose, approximately 6 ATP are consumed. The point about the 35% was like this: 11 ATP consumed in comparison to 7 ATP which is synthesized. All right. So if you take the percentage, it is approximately 35% costlier. It is approximately 35% costlier. The exact value is slightly more than 35, but it is 35% costlier in comparison to the energetics which is obtained from the glycolysis. Okay, so that is all about the gluconeogenesis. If you have no more questions, we go to the second part of the fate of pyruvate. One fate was going to gluconeogenesis, the second fate was going towards the TCA cycle. So at this point, we'll move on to the TCA cycle if you have no more questions at this point. Let us now talk about the tricarboxylic acid cycle, also known as citric acid cycle. The tricarboxylic acid, which is the first product, is the citric acid cycle, described by the scientist Krebs, so most commonly known as the Krebs cycle. Most commonly known as the Krebs cycle. Okay, the Krebs cycle. But before we reach the Krebs cycle, it will start with acetyl coenzyme A. We had said this acetyl coenzyme A is synthesized from a pyruvate by the enzyme pyruvate dehydrogenase. So first we'll look at this reaction, the conversion of pyruvate to acetyl coenzyme A, and then we will go to the Krebs cycle, also known as the Tca cycle or the citric acid cycle. The conversion of pyruvate to acetyl coenzyme A is known as the link or the bridge reaction. It links the metabolism of glucose in the cytoplasm with the Tca cycle in the mitochondria. That is why it is known as the link or the bridge reaction. It is known as the link or the bridge reaction. So in the conversion of pyruvate to acetyl coenzyme A, three things happen. In the conversion of pyruvate to acetyl coenzyme A, three things happen. Number one, addition of coenzyme A. Number two, dissolution, removal of CO2. And number three, conversion of NAD plus to NADH. Recall that all of this is occurring in the matrix of mitochondria. All of this is occurring in the matrix of the mitochondria. Okay, and the reaction is being catalyzed by the enzyme Pyruvate dehydrogenase. Here I would like to tell you that it is not a simple enzyme, rather it is a complex. The pyruvate dehydrogenase is not a simple enzyme, rather it is a complex. Alright, so conversion of pyruvate to acetyl coenzyme by the enzyme pyruvate dehydrogenase complex. Okay. Alright. Now this complex is made up of multiple components. Large number of components are there in this complex. Let's quickly take a look at the components of the pyruvate dehydrogenase complex. The pyruvate dehydrogenase complex contains three enzymes. It contains three enzymes. The first enzyme, pyruvate dehydrogenase. This enzyme is same as the name of the complex. This enzyme is same as the name of the complex. The other two enzymes are the dihydrolipoyl transacetylase, and the dihydrolipoyl dehydrogenase. The dihydrolipoyl transacetylase and the dihydrolipoyl dehydrogenase. In addition to these three enzymes, we also have five coenzymes. We also have the five coenzymes. The five coenzymes are thiamine pyrophosphate. This is written as thiamine pyrophosphate, obtained from vitamin B1. Obtained from vitamin B1. I'm sure you know what is FAD. FAD stands for flavin adenine dinucleotide. The FAD is obtained from vitamin B2. Similarly, NAD, nicotinamide adenine dinucleotide, is obtained from vitamin B3, and coenzyme A is obtained from vitamin B5. I'll write the respective names of the vitamins so that you can correlate them. B1, thiamine; B2, riboflavin; B3 is the niacin; and B5 is the pantothenic acid. So these four vitamins make important coenzymes which are participating in the reaction of the pyruvate dehydrogenase complex. Also note that the pyruvate dehydrogenase complex is similar to it is similar to two very important enzyme complexes. One is alpha ketoglutarate dehydrogenase complex, and the second is branched chain keto acid dehydrogenase complex. These are two additional enzyme complexes which are similar to pyruvate dehydrogenase complex, and therefore they catalyze similar reactions. Therefore, they will catalyze the similar reactions. So what happens? The pyruvate dehydrogenase complex will convert the pyruvate to acetyl coenzyme A, and subsequently the acetyl coenzyme A will go for the Tca cycle. If this conversion is blocked because of any reason, the pyruvate will start accumulating, and some of this pyruvate will start getting converted to lactate in the cytoplasm. So any disruption of the pyruvate dehydrogenase complex will result in lactic acidosis. Inhibitors of pyruvate metabolism lead to lactic acidosis. Inhibitors of pyruvate metabolism lead to lactic acidosis. For example, arsenite and mercuric ion react with the SH group in the lipoic acid. So they will block the pyruvate dehydrogenase and lactic acid will accumulate. Dietary deficiency of thymine. Many alcoholics are thymine deficient, both because of a poor diet and also because alcohol inhibits thymine absorption. All right. So, dietary deficiency of thymine it is commonly seen in the alcoholics. They will not consume a lot of food because they get a lot of energy from alcohol itself. They don't need extra food. But when you're consuming alcohol as the main source of energy, you're not getting the other nutrients. All right, you're only eating a little bit of snacks with it, and that absorption is also being stopped by the alcohol itself. So very poor consumption along with alcohol is a pure energy form. It doesn't contain the essential minerals and the vitamins. In addition, some individuals can have inherited pyruvate dehydrogenase deficiency. This condition is commonly known as congenital lactic acidosis. This condition is commonly known as congenital lactic acidosis. Okay, so this shows some examples where the defect in pyruvate dehydrogenase complex can give rise to the lactic acidosis. It can give rise to the lactic acidosis. Any questions so far? Any questions so far? Because now we have synthesized the acetyl coenzyme A, and we will go on to the Krebs cycle. Do you want me to repeat any part? Do you want me to repeat any part? Any part which you were not able to catch because of buffering? What part did you miss? Just let me know so that I can repeat that part. What part did you miss so that I can repeat that part? So we're starting off the Krebs cycle with the checklist. Remember, Krebs cycle is one of the three pathways which you have to know. All reactions from the Krebs cycle, the glycolysis and urea cycle are asked. So we'll definitely need to see at the reactions, but we should also know the checklist. It makes our work easier. Checklist. Point number one in the checklist, Location. Point number 1 in the checklist, location. Here I'll say all organs except with exclusively anaerobic glycolysis. All organs except those with exclusively anaerobic glycolysis. What about the compartment? Obviously, it occurs in the mitochondria. It occurs in the mitochondria. The next question talks about the substrate. Remember, when we say substrate, if you have to mark one answer, the one answer should be acetyl coenzyme A. But please note, if the option is there, you should also include oxaloacetate. Because it is a cycle, the last product will become the first substrate. So remember, oxaloacetate is also a substrate. If you have to mark one, the acetyl coenzyme A is the best answer. But if you are able to mark both, if you are able to mark both, then you go for acetyl coenzyme A and oxaloacetate. Alright. What about the end product? What about the end product? When we say end product, we end with oxaloacetate plus 2CO2 plus 3 NADH plus 1 FADH2. Again, note that we don't mention the gain or loss of ATP here. It is not counted in the end product. It is not counted in the end product. The rate limiting enzyme and target for regulation are the same. Rate limiting enzyme and target for regulation, the answer is isocitrate dehydrogenase, and this is inhibited by ATP. The acetate dehydrogenase is inhibited by ATP. So regulation by ATP. If there is more of ATP, the acetate dehydrogenase will be inhibited. The Krebs cycle will slow down. If ATP is not there, more and more of acetate dehydrogenase activity will be there, and more and more of ATP will be formed. So there is a fine balance between the ATP concentration and the rate of the cycle. And the last point, energetics. The last point is energetics. When we come to energetics, like I said, we get 3 NADH, 1 FADH2, and 1 substrate level phosphorylation, The NADH will always give 2.5 ATP because we are in the mitochondria. Will always give 2.5 ATP from NADH. There is no shuttle involved here. So we always get 2.5 ATP. The FADH2 will give us 1.5 ATP. So net result is 7.5 plus 1.5 plus 1 to give you a total of 10 ATP. The energetics for one round of Krebs cycle is 10 ATP. For one round of Krebs cycle, the energetics is 10 ATP. With this, let us quickly draw the Krebs cycle. Let us quickly draw the Krebs cycle. I'm sure you already know about the Krebs cycle, but it will be a quick recap of the reactions of the Krebs cycle. Starting with the acetyl coenzyme, which has come from the action of the pyruvate dehydrogenase, which combines with the oxaloacetate. They will combine under the influence of the enzyme citrate synthase to give you the first product of the cycle, that is citrate. The first product of the cycle is citrate. Uh, the pathway is called the citrate cycle based on the first product, and because citrate is a tricarboxylic acid, it is also known as the tricarboxylic acid cycle. Now remember that this process is occurring inside the mitochondria. This process is occurring inside the mitochondria. When there is a lot of citrate, we have the tricarboxylic acid transporter. It is a low affinity transporter. When I say low affinity transporter, it means it is able to transport the citrate only when the concentration will be high. Only when the concentration of citrate is high, the citrate will be transported outside. Outside, the citrate is quickly acted upon by the enzyme citrate-lyase. It is acted upon by the enzyme citrate-lyase and quickly converted into oxaloacetate and acetyl coenzyme. In addition, as we have seen previously, the citrate also causes feedback inhibition of PFK1. It also causes the feedback inhibition of PFK1. So if large amount of citrate is being formed inside the mitochondria, it will immediately divert some of the citrate outside. In addition, it will also try to slow down the glycolysis so that the rate of formation of citrate is decreased. All right. The oxaloacetate in turn will come back. It will come back into the mitochondria. It will come back into the mitochondria. But this acetyl coenzyme can be used for fatty acid synthesis, or it can be used for cholesterol synthesis. So remember, the fatty acid and cholesterol are also coming from the acetyl coenzyme of glucose. The fatty acid and cholesterol are also coming from the acetyl coenzyme of glucose primarily. So this is an additional point which I wanted to highlight. I'll mention this point again when we are doing the lipogenesis. Remember, the citrate which is formed in the Krebs cycle from acetyl coenzyme can be diverted outside when there is excess. When we have excess of it, it can be diverted outside. Okay, let's come back to the Krebs cycle. Let's come back to the Krebs cycle. So here the citrate has been formed. The citrate is then converted. It is then converted first to cis-acetate and then to isocitrate. First to cis-acetate and then to isocitrate. The enzyme catalyzing both the reactions is the same, aconitase. Aconitase will catalyze both the reactions. Aconitase will catalyze both the reactions, will convert citrate to cis-acetate, and cis-acetate is quickly converted back to the isocitrate. The isocitrate is acted upon by the enzyme isocitrate dehydrogenase, and then it is converted into oxalosuccinate. In this process, NAD plus is converted into NADH. NAD plus is converted to NADH. The oxalosuccinate is then acted upon by oxalosuccinate decarboxylase. Some authors will consider the oxalosuccinate decarboxylase as a part of the isocitrate dehydrogenase. So, in some of the questions or in the guides or the books, you may not find the name oxalosuccinate dehydrogenase. You may still find the isocitrate dehydrogenase. The oxalosuccinate decarboxylase may not be mentioned. Now, remember, because it is a decarboxylase, a CO2 is removed, and what you get is the alpha keto glutarate. What you get is the alpha ketoglutarate. The alpha ketoglutarate now is acted upon by the enzyme alpha ketoglutarate dehydrogenase. And remember, I told you this is a reaction similar to that catalyzed by the pyruvate dehydrogenase. So similar things will occur. What will happen? Addition of coenzyme A, removal of CO2, and the conversion of NAD plus to NADH. Conversion of NAD plus to NADH, resulting in the formation of succinyl coenzyme A. Resulting in the formation of the succinyl coenzyme A. The succinyl coenzyme A is then acted upon by the enzyme succinyl thiokinase. Now, succinyl thiokinase is responsible for the only substrate level phosphorylation that occurs here, meaning an ADP or a GDP or an IDP is quickly phosphorylated into the ATP or the GTP or the ITP, and the succinyl coenzyme A is then converted into the succinate. The succinate is subsequently first converted to fumarate, and the fumarate is then converted into malate. The conversion of succinate to fumarate is catalyzed by the succinate dehydrogenase. It is catalyzed by a succinate dehydrogenase. The succinate dehydrogenase is different from the other dehydrogenases in the pathway because it does not generate NADH. It converts FAD into FADH2. Fumarate is acted upon by fumarase, and finally malate is acted upon by the malate dehydrogenase, whereby we see the generation of last NADH. The last NADH will be generated here. The last NADH is generated here. So quickly we'll highlight the steps where NADH, FADH2 and ATP are synthesized where the decarboxylation is taking place. Isocitrate dehydrogenase catalyzes the formation of first NADH. The alpha ketoglutarate dehydrogenase catalyzes the synthesis of second NADH. The malate dehydrogenase catalyzes the synthesis of the third NADH. The succinate dehydrogenase catalyzes the synthesis of the only FADH2. The succinyl thiokinase catalyzes the formation of ATP, also known as the substrate level phosphorylation. In addition, the alpha ketoglutarate dehydrogenase and the oxalosuccinate decarboxylase also release 2CO2. They are causing the decarboxylation also. So these are the highlights of the reactions which you have to keep in mind. The aconitase will require iron. The isocitrate dehydrogenase will require manganese as the metal factor. The aconitase will require iron. The isocitrate dehydrogenase will require the manganese. Now, some of these reactions can also be inhibited. Some of these reactions can also be inhibited by non-physiological regulators. So let's take a quick look at the inhibitors of the Krebs cycle. What are the inhibitors of the Krebs cycle? We've seen the first. Let's talk about the inhibitors of Krebs cycle. Okay, fluoroacetate causes the non-competitive inhibition of aconitase. I'll talk more about this fluoroacetate very briefly, but first note down these three. Leave a bit of space after the fluoroacetate. The fluoroacetate is a non-competitive inhibitor of aconitase. The arsenite is the non-competitive inhibitor of alpha ketoglutarate dehydrogenase. Just like arsenite is able to inhibit the PDH in the same manner by blocking the SH of the of the lipoic acid, it is able to inhibit the alpha ketoglutarate dehydrogenase also. And last, most well known is the malonate. Malonate, the competitive inhibitor of succinate dehydrogenase. Don't worry about the terms non-competitive or the competitive inhibition. We'll talk about these later when we are doing the enzymes. So nothing to worry about at the moment. We'll go into the details of non-competitive and competitive inhibition when we are doing the enzymes. I'll talk a little bit more about the inhibition by fluoroacetate because this action has multiple facets. It is able to act in multiple different ways. So let's take a look at the action of the fluoroacetate. Fluoroacetate is a very important inhibitor. It is able to act in multiple ways. Let's talk about fluoroacetate. So as you mentioned, the fluoroacetate will inhibit aconitase. And if you remember, aconitase was going to act on a citrate. It is going to be acting on citrate. So when the fluoroacetate inhibits the aconitase, this conversion is blocked. This conversion is blocked. Result, the citrate level will start rising. The citrate level will start rising. And remember, when the citrate level will start rising, what will happen? Some of it will be transported outside by the TCA transporter. And there, what it will do? The citrate will inhibit glycolysis. The citrate will inhibit the glycolysis. So first point to keep in mind is directly the fluoroacetate will inhibit the aconitase. But because of this action, the glycolysis is also blocked because the citrate level will start rising. The citrate will go out and will start blocking the glycolysis. So direct action on the Tca cycle, indirect action on the glycolysis. In addition, the fluoroacetate, it combines with citrate, and this fluoride ion is released in the free form. The fluoroacetate can release the fluoride, and this fluoride can block the electron transport chain by chelating calcium. By chelating calcium, it can block the electron transport chain. All right. Also remember, the fluoride, if it goes into the cytoplasm, the fluoride will inhibit enolase. We have already seen this. Enolase is inhibited by fluoride. So again, the glycolysis will get blocked. Again, the glycolysis will get blocked. All right. Now, nowadays it is not used much, but fluoroacetate was previously used very widely as a rodenticide to kill the rats and mice in the storage houses, and very frequently there was accidental ingestion of the fluoroacetate. So this fluoroacetate in blood releases fluoride. This fluoride will chelate calcium, result sign and symptom of hypocalcemia tetany. So remember, what is the clinical presentation of toxicity of fluoroacetate? It is not the inhibition of glycolysis, it is not the inhibition of Tca cycle, rather it is the signs and symptoms of hypocalcemia, because first and foremost it is going to chelate the calcium, and it will cause the sign and symptom of hypocalcemia. So the fluoroacetate is having multiple actions. As you can see, the fluoroacetate is having multiple actions. As an inhibitor, it has a complicated effect. As an inhibitor, it has a complicated effect. All right. The second part of the Krebs cycle is the vitamins which are utilized in the Krebs cycle. The vitamins in Krebs cycle. What are the vitamins in the Krebs cycle? Now, certain vitamins we've already seen. Which are those vitamins? Vitamin B1, vitamin B2, vitamin B3, and vitamin B5. Where have we seen them? For which enzyme all the four vitamins will be required? Can you give me the answer? For which enzyme all the four vitamins will be required in the Krebs cycle? For which enzyme the four vitamins will be required in the Krebs cycle? Anyone? Anyone, can you send me the answer? For which enzyme in the Krebs cycle the 4 vitamins are required? Single enzyme for which all 4 are required. Anyone wants to give the answer? Okay, very good. We are talking about the Krebs cycle. So obviously PDH cannot be part of the Krebs cycle. PDH cannot be part of the Krebs cycle. Very good. The answer is alpha ketoglutarate dehydrogenase. The alpha ketoglutarate dehydrogenase. The alpha ketoglutarate dehydrogenase. All 4 vitamins are required for alpha ketoglutarate dehydrogenase. In addition, the vitamin B2 is required for succinate dehydrogenase. Remember the FAD converted to FADH2. B3 also required by isocitrate dehydrogenase and malate dehydrogenase. So these are the different enzymes requiring the different vitamins. The B1, B2, B3, B5 all required by alpha ketoglutarate dehydrogenase. B2 required by succinate dehydrogenase. B3 also required by the isocitrate dehydrogenase and the malate dehydrogenase. I hope this makes sense to everyone and it is clear to everyone. If you have any doubts or confusion, please ask, please get it clarified. You should not carry this confusion. Which enzyme requires which vitamin? This point should be very clear to everyone. Now I have two very simple, straightforward questions for you. How many ATP obtained? How many ATP obtained by complete how many ATP obtained by complete metabolism of acetyl coenzyme? How many ATP obtained by complete metabolism of acetyl coenzyme? Your options are 5 ATP, 10 ATP, 12.5 ATP, 20 ATP. How many ATP obtained by complete metabolism of acetyl coenzyme A? This is part A of the question. And part B of the question is how many ATP obtained by complete metabolism of pyruvate? Same options, but two different questions. Same options, but two different questions. 20, anyone else? Mm-hmm. Mm-hmm. 10 and 12.5, 12.5, 10, 10, 12.5, 12, 1.5. Very good, very good, very good. So a lot of answers. So I'll give the correct answer. For acetyl coenzyme, the answer is 10 ATP. For pyruvate, the answer is 12.5 ATP. Remember, I had said right in the beginning when we are doing the checklist, in the checklist you have written one round of Krebs cycle will give you 10 ATP. One round of Krebs cycle will give you 10 ATP, and the substrate for the Krebs cycle is acetyl coenzyme. 2 decarboxylation, the acetyl coenzyme completely exhausted, so complete metabolism of acetyl coenzyme gives you 10 ATP. In addition, we had mentioned when you're converting the pyruvate to acetyl coenzyme, one NADH is formed. This NADH is in the matrix only, so it doesn't need a shuttle. It will go to the ETC, it will make 2.5 ATP extra. So when you talk about pyruvate, you have acetyl coenzyme plus 2.5. So 10 plus 2.5, that is 12.5 ATP. The correct answer is 10 and 12.5 ATP. Okay, please remember 10 and 12.5 ATP, 10 for acetyl coenzyme and 12.5 for complete metabolism of pyruvate. Okay, so this is a little bit about the uh, TCA cycle. At this point, I'll quickly talk about the transport of glucose, and tomorrow we'll try to cover the remaining minor pathways for the carbohydrates. We'll talk a little bit about the HMP shunt. We'll talk about the metabolism of galactose. We'll talk about the metabolism of fructose, and we will discuss the metabolism of glycogen. I'm not picking up glycogen today because it will not get completed today, so I'm not picking up the glycogen because there we have the glycogen storage disorders. It will take a little bit of time, so I'll pick up the HMP shunt, the glycogen metabolism, the fructose metabolism, the galactose metabolism, and related disorders tomorrow. Today we are going to take a quick look at the transporters of glucose. In glycolysis is in Krebs cycle. Let's start from the acetyl coenzyme. When you're starting from acetyl coenzyme, you've got 3 NADH, 1 FADH2, and 1 substrate 12 phosphorylation. We had calculated this. Where's the calculation? We had calculated this. You get 10 ATP. Remember, because we have 2 decarboxylation, the coacetyl part is completely used up. In the Krebs cycle, the acetyl part is completely used up because we have two decarboxylation. Acetyl group is having 2 carbon and we have 2 decarboxylation. The acetyl group is completely used up. Net gain is 10 ATP. We have already mentioned this. We already mentioned that net gain is 10 ATP. Before that, we talked about the conversion of pyruvate to acetyl coenzyme, and here we mentioned that we gain 1 NADH. 1 NADH via the electron transport chain will give you 2.5 ATP. and you end up with acetyl coenzyme A. So when I say complete metabolism of pyruvate, that means the metabolism of the acetyl coenzyme A also. The acetyl coenzyme A will give you 10 ATP. So if you add the two, the net gain is 12.5 ATP. If you add the two, the net gain is 12.5 ATP from pyruvate. From pyruvate, the net gain is 12.5 ATP. I hope this clarifies the point. 10 for acetyl coenzyme A and 12.5 for the pyruvate. Any other questions? Any other questions before I talk about glucose transporters? Okay, let's talk about the glucose transporters. Before we go into the glucose transporters, you should have an overview of the different types of transporters. Then we come back to the glucose transporters. Let's take a quick look at transporters in general. Transporters can be passive, or rather we call it facilitative. They're not passive, they are facilitative, or they can be active transporters. All right. Main difference between the two, the passive or the facilitative transporters, they depend on concentration gradient. Why? Because they don't use ATP. On the other hand, the active transporters are independent of concentration gradient because they use ATP. This is obvious. The third point is because they depend on concentration gradient. Suppose the concentration is high in the blood, thus analyte will move from blood to the cell. If the concentration in the cell is more, then it will move from cell to the blood, meaning they are bidirectional. They are bidirectional. Whereas the active transporters are unidirectional. The active transporters are unidirectional. Okay, this is the very important difference between the facilitative and the active transporters. There's some more points that you need to remember. The transporters can be uniporters. They will transport a single analyte. Uniporters. This is mostly applicable for the active transporters. We are talking about the active transporters. They can be symporters and they can be antiporters or counter transporters. Okay. In addition, the active transporters can be primary active and secondary active transporters. They can be primary active transporters and secondary active transporters. When I say primary active transporter, it means use ATP directly. But the secondary active transporter don't use the ATP directly. What they use? They use the concentration gradient which has been established by primary active transporter. So secondary active transporter don't use the ATP directly. They use the concentration gradient established by the primary active transporter. So this is the main difference between the primary and the secondary active transporters. Now, when you talk about glucose transport, we have two important families of glucose transporters. One is the glut series. The glut series are facilitative transporters. And then we have the SGLT. SGLT is secondary active symporter. Glut series and the SGLT series. Glut series, facilitative transporter. The SGLT are the secondary active symporter. So with this background information, let's quickly go and talk about the glucose transporters. Let's talk about the glucose transporters. When you talk about glucose transporters, we are going to make a table which will have three headings. The name of transporter, the primary function of transporter, and then we'll look at where the transporter is distributed. So name, function, and location. Let's start. Okay, don't worry. I'll go back. I'll go back to the end and quickly recap where the buffering has started, and I'll repeat it. So we said that we can have the uniporter, we can have the symporter, and we can have antiporter, which is also known as a counter transporter. And when you talk about the uh, active transporters, they can be primary or secondary active transporters. In terms of glucose transporter, we said we have the GLUT series, which is a facilitative transporter, and the SGLT series, which is the secondary active symporter. The GLUT series is the facilitative transporter, and SGLT is the secondary active symporter. The full name of SGLT is sodium glucose linked transporter. So obviously here the transport of the glucose is linked with the sodium. The transport of glucose is linked with the sodium. So now that we have done this, let's go back to the glucose transporter. Here you have to make three headings. We're going to make a table. Three headings are the name, function, and a location. First, we'll write the name of the transporter that we were discussing, what is the primary function, and the tissues where it is located. Let's start with SGLT1. SGLT1. SGLT1 will transport glucose even against a concentration gradient. Will transport glucose even against a concentration gradient. The two main places where we find the SGLT1 are the small intestine and kidney. We find it in the small intestine and kidney. Although it is unlikely to be asked, I'll briefly mention that SGLT2 may be found in the kidney. The SGLT2 may be found in the kidney. So this is the extra information that you should remember, but generally you will not get questions on SGLT2. We will not go into the details of SGLT2. Now remember, SGLT1 is the transporter. It is the transporter which is helpful during a very big problem, and that is when you are having diarrhea. So that the SGLT1 is the basis for oral rehydration therapy. SGLT1 is the basis for the oral rehydration therapy. That is why we give the ORS. ORS will contain the sodium along with glucose, and because SGLT1 is still working, the sodium will move in and will take the glucose along with it. So together they are able to regenerate the electrolyte balance in the body. Energy is also provided, and the dehydration will not occur because these two particles will also carry the water along with them. The osmotic load of the corresponding two particles is replenished, and the water can successfully get absorbed even when the diarrhea is there. This is the clinical aspect of SGLT1 which you must keep in mind. The ORT is successful due to the action of SGLT1. ORT is successful due to the action of SGLT1. ORT stands for oral rehydration therapy. It is successful due to action of the SGLT1. Now let's go to GLUT. GLUT stands for glucose transporter. GLUT stands for glucose transporter. The GLUT1 is responsible for the basal transport. When I say basal transport, it means to meet the basal requirement of glucose by all the tissues. It is responsible for basal transport, so it will be found in all tissues. It is the most widespread glucose transporter. It is the most widespread glucose transporter. In addition, you should remember that all tissues will have GLUT1 plus something else. To meet their specific requirements, most of the tissues will have GLUT1 along with something else. The only exception is RBC. RBC has only GLUT1. All other tissues will have GLUT1 plus something else except for RBC. Which other transporter is there? We'll see. Gradually we'll see which other transporter is there and what need is met via those transporters. Next we go to GLUT2. GLUT2 is responsible for rapid transport of glucose. Rapid transport of glucose. So there are some tissues where very rapid transport of glucose is required. These include the small intestine, the kidney, the liver, and last but very important is the beta cells of pancreas. In all these places, the rapid transport of glucose is of very significant importance. Otherwise, the cell damage as well as the glucose homeostasis will be affected. See, from the small intestine and kidney, the SGLT will pick up the glucose from the lumen. If this glucose remains inside the cell, remember the SGLT1 is not considering the concentration, it will keep pumping the glucose inside the cell, and if you don't quickly remove this glucose, the cell will burst. So you have to quickly remove all this glucose outside the cell, and that is done by the GLUT2. At the level of kidney, sorry, at the level of liver, at the level of liver, when the glucose is reaching the hepatic sinusoids, a large amount of glucose has to be retained. Otherwise, there will be a sudden spike in the blood glucose because all the glucose which was there in the intestine has been dumped into the hepatic vein. If you allow this glucose to go into the blood circulation, there will be spike of blood glucose. So very quickly some glucose has to be withdrawn, and that is done by the GLUT2 transporter. Coming to beta cell of pancreas, you know beta cells are the cells which will release the insulin. Release of insulin depends on glucose concentration. Glucose will get metabolized. Whatever ATP is produced, that will stimulate the movement and the release of insulin. Now, if we don't have a rapid transporter, the blood glucose concentration and the beta cell concentration cannot equilibrate fast. If fast equilibrium does not occur, the release of insulin or stopping the release of insulin, both will be delayed. Alright, if the delay is there, it can be life threatening. For example, the glucose level has already risen, it is crossing 200, 300, and still the insulin secretion has not started. And when you go to the other aspect, glucose level has started falling and still the insulin is getting secreted. So severe hypoglycemia can occur and the patient may even die. So the beta cell of pancreas also require rapid transport of the glucose. Then we go to GLUT3. GLUT3 is having a very important property. It has the lowest Km. What do you mean by Km? Km denotes affinity. When I say lowest Km, what it means is highest affinity for glucose. Now this becomes important for those tissues where the glucose in the surrounding tissue is less. So wherever in the surrounding tissue we have less glucose, GLUT3 will become very important, and that is why the location is the CNS, the renal medulla, the placenta, and the cancer cells. Now see, this is very important. The surrounding fluid for the CNS is the cerebrospinal fluid. What is the concentration of glucose in CSF in relation to the blood? I'll wait for your answer. The concentration of glucose in CSF in comparison to the concentration in blood. What is the concentration of glucose in CSF in comparison to the concentration in blood? Very simple, straightforward answer. All of you must be able to answer this question. Okay, the answer is the answer is it is 65% of the blood glucose concentration. So if in the blood you have 100 milligram percent, in the CSF will only have 65 milligram percent. And we know that the requirement of glucose by CNS is more, so requirement is more and the concentration in the surrounding tissue is less. Very good, two-third is the correct answer. So we need a high affinity transporter, same for renal medulla. Remember when we are talking about aerobic and anaerobic glycolysis, at that point I had told you renal medulla receives the venous blood, so already some of the glucose has been taken out. All right, so low concentration of glucose, and if we don't have the high affinity transporter, it won't work. For placenta, the problem is slightly different. At the end of placenta, there is this perennially hungry person. Give me food, give me glucose, give me glucose. Always is making the noise. So you need the high affinity transporter to meet the requirement of the growing fetus. And lastly, the cancer cells. The cancer cells grow so fast that the blood vessels are not able to keep the pace with the cancer cells. You must remember this term from pathology. Angiogenesis lags behind tumorigenesis. Blood vessels will come later, the tumor grows in size first. Now in this case the cells are growing in size, though they are needing more energy, and in the surrounding tissue the blood vessels are not there, so obviously the glucose concentration will also be less. So until unless they have this high affinity transporter, the cancer cells cannot multiply very fast. And we use this particular feature of the cancer cells to identify the cancer cells on PET scan. We give labeled glucose, F18 labeled glucose is given, and wherever rapidly growing tumors are there, rapid transport of this glucose will occur and they will get highlighted. So we are able to quickly pinpoint the rapidly growing tumors by the action of the GLUT3. By the action of GLUT3, we are able to rapidly pinpoint the cancers on the PET scan. So you should remember this. The PET scan identifies cancer cell due to transport of glucose by GLUT3. GLUT3 will be the correct answer. By the transport of GLUT3, they are made visible. Then you go to GLUT4. GLUT4 is unique and different from all the transporters. The GLUT4 is responsible for insulin dependent glucose transport. It is responsible for insulin dependent glucose transport. And it is present in the adipocytes and the skeletal and cardiac muscles. It is present in the adipocytes and skeletal and cardiac muscles. Now here a very important point about the transport of the glucose by GLUT4. We say that insulin dependent glucose transport is there, but what you have to understand is, suppose this is the cell membrane. At the cell membrane we have the transporter, and you have this insulin. So does the insulin increase activity of the transporter? You can give the answer as yes, no. Does the insulin increase the activity of the transporter? Yes or no. We are talking about the GLUT4 transporter. We are talking about the GLUT4 transporter. Does the insulin increase the activity of GLUT4 transporter? Yes or no? Does insulin increase activity of GLUT4 transporter? The answer is no. Then what does it do? It mobilizes the vesicles on the surface of which the GLUT4 transporters are there, on the surface of which the GLUT4 transporters are there. Result, they will come up, they will come up and they fuse. Sorry, they will fuse with the cell membrane. They will fuse with the cell membrane so that the transporter is brought to the surface. So insulin actually does not activate the transporter, it mobilizes the transporter. So the correct answer is the insulin mobilizes the GLUT transporter from intracellular vesicles. It mobilizes the transporter, it does not activate. It does not activate. The activation is not a function of insulin. Why is this information very important? This information is very important because there are other factors which can mobilize the vesicle, and the most important among them is calcium. And this is the basis of the clinical advice that exercise will improve the glycemic control in a diabetic patient. See, in diabetic patient, the action of insulin is not there. Therefore, the GLUT4 which is there at the muscles and adipocytes, it cannot be mobilized. It will not get mobilized. But when you do the exercise, the calcium level will rise. Intramuscular release of calcium. This calcium will mobilize the GLUT4, and this GLUT4 can then start moving in the glucose from blood into the cell. So remember, the mobilization is by insulin, not activation. Activation is not required. Once it is on the surface, it will act. Once it is on the surface, it will act. So you have to make sure that the transporter reaches the surface. So we can do this also by exercise. The calcium will mobilize the GLUT4 and ensure that it is there on the surface. That is why exercise improves in glycemic control in type 2 DM patients where insulin resistance is there. Insulin is there, but it is not able to act. So its action can be simulated by exercise by releasing the calcium. The last in the series, and that is GLUT5. The last in the series, and that is GLUT5. This time my screen display has gone. Let me see. Okay, just give me a second. Okay, so we come to the GLUT5. GLUT5 is responsible for transport of fructose in intestine. It is present in the small intestine. Remember, it is not present, I'll highlight, it is not present in kidney. It is not present in kidney. The GLUT5 may also be seen, it may also be seen in the seminal vesicles, in the testis. It may also be seen in the testis where it helps in transporting the fructose inside the seminal vesicles. Okay, now once you know this, you should also know the transport of glucose across the intestinal epithelium, because a lot of questions will be asked from that particular scenario. So I'll quickly draw the transport of glucose at the level of intestine. So start thinking about any doubts or topics you want me to repeat, because after that we'll be signing off for today. Okay. So this is the okay. You're right. The networks are also getting hypoglycemic. So what I'm going to show you is the transport of glucose at the intestinal epithelium level. So we have two surfaces, as you know. One is the luminal surface, and the second is the basolateral surface. The second one is the basolateral surface. On the luminal side, we have the intestine. We have the intestine, and on the basolateral surface we have the blood vessel. We've just started the transport. We have not done anything, don't worry. We have not done anything about the glucose transport in the intestinal epithelium. I'm just drawing the diagram. Okay, this is the blood vessel, and this is the intestine, right? Okay, so first and foremost, we know in all the cells we have a very important transporter. In all the cells we have a very important transporter which will move the potassium inside and will move the sodium into the blood. Which is this transporter? Which is this transporter? Sodium potassium ATPase. नीचे sodium potassium ATPase. So as a result, what is happening? The concentration of sodium inside the cell is decreased. You can see the sodium is moving outside, so the concentration of sodium inside the cell is decreased. This is the primary active transport. It is a primary active transport, and it is creating a concentration gradient. This concentration gradient which is being created will be used by a secondary active symporter. It will be used by a secondary active symporter, with the help of which the sodium can now be brought in. The sodium can now be brought in. Along with this sodium, we are able to transport other molecules. In this case, it is the glucose and galactose. The glucose and galactose will come in. Which is this transporter? The name of the transporter? It is the sodium glucose linked transporter. The sodium glucose linked transporter The SGLT1 will transport the glucose and galactose along with sodium. Remember, it is the secondary active symporter. It is using the concentration gradient established by the sodium potassium ATPase. Please note, SGLT1 does not transport fructose. SGLT1 does not transport fructose. So how will we transport fructose? The fructose is transported by the glut series. The only glut which is there on the luminal surface is the glut 5. Only glut on the luminal surface is the glut 5. So this glut will transport all glucose, galactose as well as fructose. So this means the transport of fructose from the lumen to the cell is carried out only by the glut 5. The SGLT will not transport it. So what we get? We get the fructose also. Now, due to action of the SGLT, rapid collection of the glucose galactose is occurring inside the cell, which must be quickly moved outside. So now what do we need? We need a rapid transporter. We need a rapid transporter for all these. Otherwise, the cell will burst. We need a rapid transporter for all these, and we know that rapid transporter happens to be GLUT2, which will quickly push out the glucose, galactose and fructose from the cell into the blood. It will rapidly push out the fructose, glucose and galactose from the cell into the blood. It is the rapid transporter. Alright, so I'll quickly summarize. The primary active transporter in this picture is the sodium potassium ATPase. Please note, the sodium potassium ATPase does not transport glucose. It is not a glucose transporter. So if you ask you about glucose transporter, sodium potassium ATPase is not included. It is a transporter which is there on all the cells. It is not involved in glucose transport. It is facilitating the whole mechanism, but it is not a glucose transporter. Remember, sodium potassium ATPase is not a glucose transporter. The SGLT1 and GLUT5 are present on the luminal surface. SGLT1 and GLUT5 are present on the luminal surface. The GLUT2 is present on the basolateral surface. GLUT2 is present on the basolateral surface. The SGLT1 is the secondary active symporter. It will only transport glucose and galactose but not fructose. The GLUT5 will transport glucose, galactose and fructose. Therefore, it is the only transporter of fructose on the luminal side. The fructose, glucose and galactose from the cell are pushed into the blood by the GLUT2. They are pushed into the blood by the GLUT2. Exactly same arrangement will be there in the kidney. Only thing is, in kidney, I had mentioned, in kidney no GLUT5. Therefore, whatever fructose has been filtered in kidney cannot be reabsorbed. Filtered fructose not reabsorbed in the kidney. Filtered fructose not reabsorbed in the kidney. So you have to remember these points that I've highlighted here. On the basolateral surface we have the GLUT2, on the luminal surface we have the GLUT5 and the SGLT1. If you have to identify one transporter which is involved in glucose transport, you will go for the SGLT1. It is the most important transporter. Alright, but this is the complete picture which you have to keep in mind. So we'll be stopping here. Tomorrow we'll start with the HMP shunt and then we'll go on to the glycogen metabolism associated disorders, and finally we'll talk about the galactose and the fructose metabolism. If you have any questions, I'll take them now. Else we will call it close for today. Any questions? I'll wait for 2 minutes. If you have any questions, any part you want me to repeat, feel free to mention them. No questions? If there are no questions, just give me a thumbs up so that I know that the system is working, you are able to hear me, and you are able to communicate. If there are no questions, just give me a thumbs up. If you want any topic to be repeated, or if there are any doubts, just write them down. No questions? If there are no questions, then we'll close this session. If there are no questions, we will close this session. So that will be all for today. And tomorrow, same time, we'll continue when we'll pick up from the HMP shunt. The glut will transport all the hexoses, whether it is glucose, galactose, or fructose. Glut2 will transport fructose. Yes. Glut1 will be there on the surface from where the blood will provide the glucose. So it will be there on the basolateral surface. Glut1 will always be facing the blood. Glut1 will always be facing the blood. Glut1 will always face the blood. No, citrate cannot promote gluconeogenesis. Citrate will not cause the gluconeogenesis. Citrate cannot cause the gluconeogenesis. Okay, I'll quickly recap the transport in the intestinal epithelium and then we will close for today. Okay, so the gradient for sodium is generated by the sodium potassium ATPase. As a result, we have low sodium concentration in the cell. This is utilized by the secondary active symporter SGLT1. SGLT1 will bring in the sodium along the concentration gradient, and the glucose and galactose will hitch a ride because it is a symporter. They'll hitch a ride and they will reach inside the cell. The GLUT5 is also there. The GLUT5 is able to transport glucose, galactose, and fructose, all of them. So the concentration of fructose, glucose, and galactose will start rising in the blood because SGLT1 is concentration independent. So that there is no damage to the cell, the rapid transporter GLUT2 will transport the fructose, glucose, galactose outside into the blood, and from there they can go to the rest of the body. Similar arrangement will be there in the kidney. Only difference will be in the kidney the GLUT5 will not be there, so whatever fructose is filtered will not be absorbed in the kidney, but in the intestine whatever fructose is there in the diet can be taken up